Inside pretty much every single cell in your body, there's this incredible instruction manual that, well, it makes you you. It's your DNA. But have you ever wondered how life makes a perfect copy of this ridiculously complex blueprint? Today, we're diving into DNA replication, and let me tell you, it is nothing short of a masterclass in molecular precision.
And that's really the big question, isn't it? Every single second, millions of your cells are dividing. And before they do, they have to run this tiny genetic copy machine. And they have to do it flawlessly. So how on earth does this microscopic machine actually work? Well, let's find out. So first things first, let's talk about the why. Why is all this copying necessary? Well, think about a tiny seed growing into a huge tree, or even just your own body healing a small cut. All of that is happening because of cell division. And every single time a new cell is born, it needs a complete, perfect copy of that DNA instruction manual. And I really can't stress this enough. The copy has to be perfect. We're talking about a code that's 3 billion letters long. Even one tiny mistake can have huge consequences. So this need for precision, it's not just some minor detail. It's literally the entire point of the process. All right, so how do you even start a copy job this important? It all kicks off by getting the DNA molecule ready. And the process kind of looks like unzipping a jacket. This is where our amazing molecular machinery gets to work. To wrap our heads around all this, we're going to break it down into 4 main phases. It's pretty straightforward. First, we have to unwind the DNA. Then we prime it, kind of like getting a wall ready for paint. The main event is next, elongation. That's where the new strands are actually built. And finally, we join all the pieces together. So let's meet the team that gets all this done. Now here's the really cool part. The cell uses this super specialized toolkit of enzymes to pull this off. You can think of them like a construction crew. First, we've got helicase, our unzipper. Then there's primase, the starter. The star of the show is DNA polymerase, the master builder. And cleaning everything up at the end is ligase, the molecular glue. So, kicking things off, we have helicase. Just think of this enzyme as the unzipper. It literally zips along the DNA, breaking the bonds that hold the 2 strands together. And as it opens up the DNA, it creates this Y-shaped area that we call a replication fork. This is what exposes the genetic code so it can be read and copied. But here's the thing. Our master builder, DNA polymerase, can't just start building on a bare strand of DNA. It needs a starting block, a green light. That's where primase comes in. This enzyme is the starter. It lays down a little piece of RNA called a primer, which basically shouts, okay, start building right here. Okay, so the DNA is unzipped and the starting blocks are in place. Now we're ready for the main event, the actual construction of 2 brand new strands of DNA. And the star of this show, without a doubt, is DNA polymerase. This enzyme is just incredible. It cruises along each of the original DNA strands, reads the code, and grabs the right matching nucleotide, the A's, T's, C's, and G's, to build the new strand. And get this, it is unbelievably fast and accurate, adding around 50 of these building blocks every single second. Now, this is where it gets really, really clever. See, DNA polymerase is a bit picky. It has a strict one-way rule. It can only build in one direction. For one of the strands, which we call the leading strand, that's no problem at all. It's built in one smooth, continuous piece. But the other strand, the lagging strand, runs in the opposite direction. So what do? Well, you have to build it backwards in a bunch of short, disconnected chunks. And these short, separate pieces of DNA on the lagging strand, they actually have a name, Okazaki fragments, named for the scientists who figured this all out. It's a really smart, if kind of messy, solution to that whole one-way street problem. So now we've got this one strand that's all chopped up into these little Okazaki fragments. How do we turn that into a complete, unbroken strand of DNA? Well, that's the job for our final enzyme, ligase. It comes in and acts like molecular glue, sealing up all the little gaps between the fragments to create one single unified strand. And with that last little bit of glue put in place, the high-speed construction job is finally done. The unzipping, the priming, the building, the joining, it's all complete. And what's the result of all that incredible work? Well, from one original DNA molecule, we now have 2 perfect, identical copies. This is the entire point of the process, making an exact duplicate of the genetic blueprint. So what we're left with are 2 identical DNA helices. And here's a really elegant detail. The process is called semi-conservative.
Now, that's just a fancy way of saying that each of the new DNA molecules has one of the original old strands and one completely new strand. It's a perfect mix of old and new, ensuring nothing gets lost. And just like that, the blueprint for life is copied, checked, and ready to be passed on to a new daughter cell. This entire beautiful, intricate dance ensures that the instructions for life are carried forward generation after generation after generation.
I mean, the precision is just mind-boggling, with an error rate of less than 1 in a billion. But it's not absolutely perfect. And that one simple question, what happens when there's an error in the code? Well, that opens up a whole new universe of genetics, everything from evolution to disease. Definitely something to think about.